All right, everybody, I'm going to go through the week 3 review. This is an overview of chapters 7, 10, 24, and 25. And there's also 11. It just doesn't say this on the front of this, but yeah, we're going to be going through all of those today. It isn't as bad as it sounds. So let me do this. All right. All right, first is chapter 7. Chapter 7 deals with microbial nutrition. So our first question for this chapter, compare and contrast heterotrophs and autotrophs. So this is talking about essentially where these organisms are getting their carbon sources from. A heterotroph is somebody that gets their carbon source from essentially breaking down another organism and using its organic molecules for its own. And then autotrophs are the ones that are going to be getting their carbon sources essentially from inorganic sources and fixing them into organic molecules. The autotrophs are able to do that thanks to typically either using energy from the sunlight or other mechanisms. We're talking about some of the more less common ways that organisms can do this. But anyways, the point is they fix the inorganic stuff into organic molecules. And whenever the heterotrophs go and eat the autotrophs, they just steal those organic molecules essentially from their structures and all that. So that's how that works out. So why do organisms need each of the following elements? We have CHOMP, which is our little acronym here. This is CHONPS. This is referring to carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. And basically, I did not know there was a dog behind me. Sorry. I like what the translation says as well. Dog bowling feet. Dog behind me. So hi. Anyways, got distracted by the dog. Carbon, of course, we know this makes up most of our organic molecules within our cells. Any biochemical macromolecule is based off of carbon with a hydrogen backbone. So of course we need hydrogen. Nitrogen is important in part of our nitrogenous bases in our nucleic acids. And then phosphorus, of course, that's going to be incorporated as phosphate within a lot of our nucleotides as part of our nucleic acid structure. And if we think about ATP, ADP can't become ATP without, the addition of phosphates. And there's already phosphates in that too. So we need phosphorus for that. And then sulfur, we don't think about much because we don't talk about it much, but it's incredibly important as far as the structure of our proteins and maintaining the structure of our proteins because it will be found in some of the amino acids that are able to associate with one another and in disulfide bonds. Okay. All right, so what types of organisms form the basis of most food webs? That's going to be the autotrophs. And typically, most commonly, most of them are going to be the photoautotrophs, which means photoautotrophs are getting the energy that they need to fix those inorganic carbon molecules into organic molecules as autotrophs, right? They're getting that energy from the sun. So those are photo for the light and autotrophs for creating, their own organic molecules using that energy from the sun. So they are usually the basis. We call them primary producers. That's an important term to remember. Primary producers produce their own, organic molecules. They're primary because they have organic, because they take inorganic carbon dioxide and turn it into organic molecules. And because they're going straight from just energy from the sun, they don't have to get it from other organisms. Everything is like from the start, from scratch, from get-go. So primary producers. All right, compare and contrast endoparasites, ectoparasites, intracellular parasites, and obligate parasites. Endoparasites are parasites that live inside of your body. Ectoparasites are parasites that live outside of your body. So it could be like the ecto might be like ticks or even lice, stuff like that. The endoparasites, these are going to be things like the worms that you could get in your something like tapeworms and everything like that. These are the obvious ones, I feel like, because you can technically say that a lot of the bacteria that cause disease or the viruses that cause disease, we refer to these as parasites as well, because they are having a relationship with our bodies that is necessary for their survival, but it is causing us harm. And that's what parasitic means. So just think about that way. Intracellular parasites are parasites that live inside of the cells. And obligate parasites are parasites that have to be parasites and they cannot live any other way. Sometimes we have parasitic relationships with some organisms that aren't always parasites, right? So like if we were talking about E. coli that normally lives in our colon and functions to break down our foods and does normal E. coli colon stuff, when it moves into the urinary tract, then it is down outside of its little area. It can cause problems and that can develop into a parasitic relationship. So it's not always parasitic, right? Okay. Explain diffusion and osmosis. Diffusion is just movement of molecules from an area of high concentration to an area of low concentration. So my little analogy always is you have a pile of balls held up by this net. When you let the net off, you take the net off, the balls are just going to want to roll everywhere and be kind of evenly distributed everywhere until, you know, they settle. That's diffusion. The same thing with molecules. Okay, they just want to sort of relax and be spread out and even everywhere. With osmosis, we are talking about the movement of water molecules that way. So the diffusion of water molecules is referred to as osmosis. That brings us into those concepts of isotonic, hypertonic, and hypotonic. And isotonic means that what's going on inside of the cell with the concentration of solutes, so the stuff that is dissolved in basically, for our living purposes, water, right? That's the number one solute, solvent in life that's going to be dissolving all the solutes like salts and things like that. So our cells, inside of our cells and our cytoplasm, what is that noise? In the cytoplasm, there's like nutrients and electrolytes and all this stuff. So it's not just pure straight up water in there, right? So there's all sorts of stuff dissolved within that cytoplasm, but it is mainly made up of water. It's water-based. So that's our solvent here. So you take what's inside the cell, the solvent being water, the solute being what's dissolved in it, and take outside of the cell. And then we're comparing the two. And the outside can be isotonic, which would be the same solute concentration outside as inside. Okay. Hypertonic. Hyper, just think of hyper being a lot. So hypertonic, outside of the cell is going to have more of the solute. So things like salt would be higher concentration outside than what is inside. So if there's a whole bunch of salt outside of the cell, what's going to happen inside of the cell? We can't move the salt across. Okay, membranes are selectively permeable, so the water can get across, but the salt can't. So how are we going to equal those concentrations out? Well, the cell starts pushing water out of itself to try to make the concentration more diluted outside and more concentrated inside by moving the water. And that's by osmosis, diffusion of the water. So it'll try to make it equal. If there's a whole lot of salt on the outside, of course, it would not necessarily be able to do this. So the cell will shrink down potentially to a deadly point. That's called plasmolysis or plasmolysis, however you want to say it. I don't care as long as you can recognize the term. And then we have a hypotonic. This is whenever the salt concentration or solute concentration is lower outside. So hypo being low, So it's lower outside the cell than what is inside of the cell. So essentially what's going to happen is water from out, remember water moves, not the salt, only the water. So with the water from outside moves into the cell to try to make the concentration inside of the cell the same as what is outside, to try to equilibrate. And so when it is doing that, that will cause the cell to swell up. So if you put a cell into fresh water, that's just straight up fresh water, this is what will happen. The cell will take up all that water because we have all these salts and stuff inside of the cell, and that will cause the cell to swell. Bacteria with cell walls are able to maintain their cell shape and not burst. So the cell wall helps the bacteria with that. Of course, gram-positive cells are going to have thicker cell walls and are more able to maintain that than the gram-negatives. Cells that have a poor cell structure that aren't as strong as that cell wall, they will burst when they fill up too much. That makes sense. Okay. Selective permeability, we just mentioned that. So that is going to be the fact that the cell membranes will allow the passage of water and certain sort of non-polar molecules to cross up, very small ones, to cross across the membrane freely, but everything else can't. Nothing else can do it on its own, just across the membrane itself. Because remember, we have those charged phosphate heads and those hydrophobic tails. So that affects the ability of these molecules to move across the membrane. So that's essentially what's going on there with being concerning there. And so facilitated diffusion, that's just diffusion. So diffusion is still moving from an area of high concentration to low concentration. So the way that it wants to move, that's just how things want to be. So facilitated diffusion allows diffusion to happen, but now we're putting like channels and things like that into the cell membrane to allow diffusion of these other molecules that can't cross the membrane just straight up by themselves. These channels will allow them to move across from high concentration into low concentration, how they want to move. This doesn't require any energy. If you want to move something against a gradient, which is our next question, number 9, then you will need to put energy into it, because that's not going the way that, you know, the laws of thermodynamics want us to go. So we have to defy the laws of thermodynamics, not really defy them, but make an exchange. So we put energy into the system to allow us to move something against a gradient or at a faster speed than what is natural. So that would be active transport that utilizes channels that will move things against the gradient, but will require the use of energy to do so. Active transport. So what is the difference between phagocytosis and pinocytosis? Phagocytosis can be large engulfing of usually matter, cells, things like that, that we're going to be digesting often just to get rid of, you know, damaged or dead cells or to destroy any potential pathogens that we come along. Then pinocytosis is like what we call cell drinking. So it's taking little sips of typically liquids that are in the environment, like oils and things like that. So the cell will take up little sips from its environment to incorporate it and to use it for whatever it's using it for in the cell. That is pinocytosis. I always remember it. I think of like a Pinot Noir or Pinot Grigio or something like that. Well, you do. You hopefully aren't chugging your wines, but I hope you're not living that life. So, anyways, the differences, but the different terms for the groups of organisms based on their temperature preferences, not particularly exciting here, right? So, the psychrophiles, those guys live preferentially in the cold. We have the psychotrophs that can live in the cold, but don't necessarily prefer it. We have the mesophiles, which are like our room temperature and pathogen type of guys. So all the pathogens fall into that category. And then we have the thermophiles that like the higher temperatures. And we have the extreme thermophiles like the archaea that live in like extremely high temperatures that could be even flourishing above boiling temperatures. There's another term called thermoduric, which means that the organisms will be able to tolerate higher temperatures for short periods of time, but they don't prefer to be at those higher temperatures. They just can tolerate it. So those are important terms. Next thing, what impact do oxygen byproducts have on microbial life? Oxygen byproducts and related toxic oxygen ions and compounds. These guys are toxic to cells. They can destroy molecules and cause damage to cells. So organisms that have to be exposed to oxygen or use oxygen as part of their metabolism, which was explained in the metabolism chapter in chapter 8, which hopefully you've already read. Chapter 8 tells us that aerobic respiration requires oxygen as our final electron acceptor. And during the whole process of dealing with that and making water from the protons and the electrons and the oxygen molecules, we can have the creation of these toxic byproducts as well. And they also just exist in the oxygen environments. So some cells that are not adapted to being in oxygen can be pretty severely damaged by these toxic products, and they don't have the means to deal with them in a way to keep themselves safe from harm from these things. Anything that uses oxygen or lives in oxygen will have to have some sort of mechanism to deal with this. Otherwise, their cells will get destroyed and they'll die. So what is the term for microbes that live in high salt environments? We call these halophiles. What is unique about some of these? Some of the halophiles actually require high salt concentrations to live. And that's because they've adapted themselves to bring salt into their cells. So when we're talking about the comparison of outside and inside the cell and the tonicity, whether it's iso or hypo or hyper, they will equal that out by pulling salt into their cells so that if it's high concentration outside, it'll also be high concentration inside. And that makes it isotonic for them in that environment. Now, if you take them out of that environment, they'll die in an environment where our cells would normally survive just fine because it's not enough salt and it'll cause them to essentially, burst. So everything will be hypotonic to them at that point outside of that. All right, what is symbiosis and what are the three major kinds? Symbiosis is whenever there are two organisms that are in a close living relationship where at least one of those organisms depends on that relationship for survival. The three major kinds, we have mutualism, where both organisms benefit. Commensalism, where one organism benefits and the other one is neither harmed nor helped, just is there. And then we have parasitic, where we have one organism benefiting from an act, at a cost to the host, which is going to be damaged by the relationship. So we know how that would work. So it's kind of bad for one guy, good for the other guy. How do microorganisms and biofilms monitor the overall health of the biofilm? They sort of communicate with one another with chemical signals. They'll send out certain chemicals that notify other organisms in that biofilm that things are going good, or that, we're running out of this nutrient or whatever it is, and can help maintain the overall health of that biofilm using that system. And that whole process where they're communicating with one another with those chemical signals in the biofilm is called quorum sensing. All right. Explain the phases in the bacterial growth curve and what's happening in each. What are some methods microbiologists use to monitor this? All right. First of all, if you want to know if your cells are growing or if they are there are cells there, that's a different thing. Those are different things. You want to know if your cells are actually alive versus that there are cells there. Sometimes you can look at your tube and see that it is cloudy, that is turbid. That's the term for that. And you'll say, oh, there's cells in this. This is definitely a good culture. It's growing, it's healthy. But what if they all died already and they're all just dead in there? It's still cloudy. So there's a problem there. So how are you going to actually, be looking at that sort of stuff? So there's different techniques you can use to determine if your cells are alive. But probably the most simple and common way to do this would be, a viable cell count or viable cell plating. And what you would do is you would take samples at various time points if you want to know how healthy the population is at that point, and take a sample of it or a dilution of that sample and plate it and then count the colonies. And that should correspond over time to the health of that population that you're looking at. So then you're looking at live colonies. They have to be alive to make colonies. So you're looking at live organisms, and that can give you a sense of living organisms versus not living. So, all right. So that's how you might monitor something like that. You can also use something called flow cytometry that will use a laser and certain markers and measure the size of the cells. And it basically has different ways that it can measure the cell to see how healthy it is, and even determine if it is alive or not, and looks at each cell one by one. So it's a pretty useful tool for examining that if you need to know more detail about your population than just alive or not. You can measure turbidity of cultures, which would be putting it into a spectrophotometer, where you take your little cloudy tube and you put it into a machine that shines light through it, and then it measures the amount of light that gets through, and that tells it how cloudy it is and can base how many cells are in there. You might just want to, assume that your cells are living. It depends on what you're doing, of course, right? What do you need to know about your population? So the phases in the bacterial growth curve, we have the lag phase. That's just the phase where we just inoculated the sample. If you just inoculate your sample and you just put it into the incubator, like you spread all your stuff on your plate all over it to create the lawn for a Kirby-Bauer assay or something. It actually caught that in the translator, so that's kind of impressive. Anyways, Kirby-Bauer, you make your little lawn of your bacteria with the swabs, right? You streak it and turn it, and then streak it and then turn it, and then streak it and then turn it, and then bacteria will be all over the place. But when you were streaking it and you went and put it into the actual incubator, do you think that if you, counted how many new cells were there 5 minutes later, that there would be just a whole ton of them? No, because you just put the bacteria there. They're just getting ready to grow. They're just, gathering everything they need to start dividing, to start replicating their genomes in order to create new cells and everything like that. That takes a little bit of time. So that's the lag phase. The log phase is the next step. It's also known as exponential growth, or the exponential phase. And basically what is going on there is we have exponential growth. Well, how do you want me to explain that better? Their cells are going to be doubling and then doubling and then doubling and doubling at maximum rate, hopefully. And that's going to lead to the fastest growth rate that you're going to see in that experience. So that's the log phase, a logarithmic phase, or the exponential growth phase. Next is the stationary phase. This is whenever growth has sort of slowed down. There's not as much new stuff available. We don't need to make as many new cells because the population is reaching a certain point. Not as many resources are available because of so many cells are there. And we start having die off of some of the cells where growth stops or slows, and growth may equal death at that point. So we have a plateau there in the population. And once we start having the cells no longer have nutrients, there are no longer, having a healthy environment in which to live because there's waste products from other cells dying and all of that sort of stuff, then they start dying off at an exponential rate as well. So just in the opposite direction. So that is how that works. So that is it for chapter 7. So I'm going to go ahead and move on to chapter 8. Nope, I lied because I forgot we're not doing these in order. We're going to move on to chapters 24 and 25. I forgot we already did chapter 8. Sorry, I've been recording so many videos. Okay, Chapters 24 and 25 are dealing with like environmental and applied microbiology. You can see there's not a ton on this screen. So the biosphere, what are the three realms of the biosphere? The biosphere is any portion of the planet that can support life, all of it. Any of it that can support life combined together represents the biosphere. The three sort of realms that we look at for the biosphere, that's going to be the lithosphere. The lithosphere is the like earth. There's going to be the hydrosphere, which is the water, obviously. And that's an environment things can live in. And then we also have the atmosphere. Go figure what that is. It's the air. So organisms can exist in that. It's maybe not necessarily flourishing in the air, but it can be an area where organisms can be alive. So what is a biome? A biome is like area in the biosphere as part of the biosphere that has a certain climate and type of flora and fauna and that sort of a thing. So that, yeah, like the rainforest, you guys know this, the savanna, the desert, those are biomes. So they have a climate that is unique to them that lends to specific organisms being able to survive there and flourish there. The community is a group of organisms that are living close to one another and whose lives may affect one another. All different kinds of organisms, whereas the population is talking about a single species of an organism in that community, typically. So talking about, you know, the E. coli, you know, why am I always using E. coli as an example? I know the spider monkeys in the rainforest, okay? That would be a population. Okay, so that's the differences between those. Energy flow within a food pyramid. We sort of touched on this in the last chapter, but basically the photoautotrophs are our primary producers. They are at the base of the food pyramid because they create their own energy, essentially, from the sun. And they use that energy to take inorganic carbon dioxide and put it into organic molecules, right? So we learned a little bit about that with photosynthesis. So those guys are our primary producers. And we've already explained in the last slide why we call them primary producers, right? They are the from scratch guys. They are not stealing any of this from any other organism and turning it into other types of molecules that higher up organisms in the food pyramid, the consumers, which rely on the producers for, you know, food, carbon, energy, and other nutrients. The consumers will rely on eating those producers. So the producers are at the bottom. They're producing the energy that will be used by the entire sort of ecosystem. The higher up you go in a pyramid, you lose energy at each step because some of the energy that I get from eating a plant will be lost either as heat from often metabolism, just from the whole process, or kept maybe to build my cells, but it can't be regained by the next organism that might eat me, for example. So you'll lose energy at each step. So things at the top of the pyramid, those primary apex predators, the apex predators are going to have to eat more mass of whatever they're eating below them in order to get the appropriate amount of energy compared to what things at lower tiers of the food pyramid would need to do. Carbon and nitrogen cycles and climate change. I mean, I have a little bit more detail of the nitrogen cycle here, but you guys pretty much know the carbon cycle. There's carbon dioxide in the atmosphere that photosynthetic organisms can utilize via photosynthesis by converting that carbon dioxide into organic molecules like glucose, which they'll use for energy, but they'll also be consumed by other organisms, the heterotrophs. And the heterotrophs will utilize those organic molecules that they made as their carbon, and then move on down the food chain. Then we also have the saprobes, and the saprobes do essentially that same thing. They're breaking down dead matter. Dead detritus is the term for that. So they'll break apart the detritus and break it down and use all of its components for their own cells. All of these organisms, the heterotrophs and the saprobes, all of the other heterotrophs that are in between all those steps, those guys are going through aerobic respiration. And they are releasing carbon dioxide as a waste product during aerobic respiration. And so carbon dioxide gets released back out into the environment. We're also having a release of carbon dioxide into the environment from the environment itself. In other words, from things like volcanoes erupting or from industry, of course, we know this, or cars or combustion in general, man-made stuff that releases carbon dioxide. Then we start to cycle back over again with the photosynthetic organisms. So that is how the carbon cycle works. Now, the nitrogen cycle is a little bit trickier. We could go into the little differences of it if we wanted to, I guess. When we talk about nitrogen fixation, we're talking about bacteria in the soil that can take nitrogen from around it in the environment, in the soil, and convert it into a usable form for the plants that they are in a symbiotic relationship with. Plants depend on this in order for them to survive. So if bacteria cannot survive in the root nodules that they are occupying and provide nitrogen to the plants, then the plants won't survive. And that will lead to depletion of plants on the planet. And that is happening in real life due to global warming. So that's nitrogen fixation and that part of it, and the global climate change aspect of that. Carbon dealing with combustion and the issues related to that. Nitrogen relating to plants and nitrogen fixation and that sort of stuff. So there are other nitrogen cycles. There is ammonification, there is denitrification, and there is nitrification. So there's three other ways that organisms can handle nitrogen, having to deal with converting it into different forms, whether that could be ammonia or nitrite or nitrate or nitrate or intermediates like that. And in the denitrification, we would be talking about even creating nitrogen gas, N2. So, that's different ways that it can be cycled in the environment, but ultimately, it's just like any other cycle, and it's just going to be reused over and over again, depending on the organisms that are utilizing that version of nitrogen or carbon. So potable refers to water that is free of pathogens. It doesn't have to be completely pure. No, it doesn't have to be completely clear. No, it has to be free of pathogens. Doesn't have to taste nice. The palatable would be nice. We are not talking about palatable. We are talking about potable. Comes from the Latin word potare. So FYI, that's why it's called potable and not potable. There's a thing there. So we're going to explain the steps of drinking water treatment and the basic steps of wastewater treatment, and of course, why that's important. So first, I'm going to pull up my little notes on this. Okay, because I don't want to miss that part of it. We talked about these. Drinking. So yeah, here's the steps that I do want to mention to you guys. Okay, drinking water. When we are filtering drinking water to make sure that it is safe for consumption by people who are getting it out of their taps at home, we start with sedimentation of whatever our starting water is, usually from an aquifer, like from a well, something like that. And we're going to go put it through sedimentation, removing large things. Basically, it's like filtering large things out and letting the things that aren't water settle out of the water and taking the water separate. Next, we will aerate the water, actual fluid portion that we've kept from sedimentation. The fluid portion will get aerated. Why would we want to do this? Can you guys think of a reason why? Because we just talked about it in the previous slide. It has to deal with exposing microbes to those toxic oxygen compounds. A lot of organisms that cause disease that are going to be in water will be sensitive to that sort of a thing. Not all of them, but we can eliminate quite a few of them just by exposing them to oxygen. So that's what aeration is for. Then we have filtration, which is pretty straightforward. We're actually filtering now the water that we have treated with the aeration, like you would filter anything. And then we are going to treat it with chemicals like chlorine and ozone, and then store it until you are ready to consume it, like in a water tower or something. All right, the wastewater is a little bit different. There's more steps because there's more junk in the water, of course. So here we are talking about sewage treatment. The primary treatment of sewage is going to involve removing the large matter. Part of this will be sedimentation. Part of this is going to be what they call skimming. We're just trying to remove any sort of solid matter that can be quickly, separated out from what is left. Once we've removed as much of that obvious solid matter as we can, we will now go through the secondary treatment, which is going to involve biodegradation of sludge first. I know it sounds disgusting because it kind of is, but it's like the semi-solid material that's not quite water, but it's not quite removable, like the other actually solid stuff, like what you would imagine, if you had a bicycle in there or something that's obviously solid, right? Obvious solids. Well, the sludge is the stuff that doesn't quite make it as far as solid or liquid. It is, poopy stuff, fecal matter a lot of the time that could be left over and just incorporated in with the water itself. So we have special bacteria and protozoa that can break down that sludge using enzymes that they possess naturally, and use those to break down the sludge so we can gain even more water from this process. We can save as much water as possible. So that's biodegradation of sludge. Then we have aeration and filtration again, just like we did with the drinking water. And then finally, our tertiary treatment will include more filtration steps, because again, we need to really filter this out. And then we have treatment with chemicals, just like we did in the drinking water, whether it's chlorine or ozone or whatever it is. And then we will return it back to the environment then. So instead of children playing out in the rivers where you're just dumping waste, literal sewage into the rivers where they are washing their clothes, where they're bathing, whether they're playing or even getting their drinking water from there. Now we can treat that and give it back to them in a much safer, much cleaner form so that we are not spreading disease that would be spread within fecal matter and stuff like that. So that is a very useful, thing that we've developed over time and need to incorporate into more less fortunate areas of the world. Coliforms, good, that's good. So coliforms are basically indicators of whether or not there's poop bacteria there. This is going to include E. coli, and it's probably the most important of the coliforms that will come from the colon, be present in sewage. And if your treatment is going correctly with your drinking water or with your reclaimed sewage water, oh my God. Oh, I thought I turned this off. That scared me so bad, you guys. Okay, you'll be watching this later and be like, I remember that day. Yeah. Heart attack. Okay. So anyways, we will do testing and plating and, filtering, and then putting the filter of the tap water or your reclaimed sewage water, whatever it is, and putting it onto actual media like plates and letting it grow and then counting the colonies on there. And the colonies that are associated with poop, with fecal matter, and that if they are present in the sample, that indicates that there could be other pathogenic microbes like salmonella or other things that could be present there as well. So that is what this would be looking for. So if you have E. coli there, you might have salmonella, shigella, cholera, all that sort of stuff. That's what coliforms are. They're little indicator bacteria. They are fecal coliforms. The fecal bacteria are there. The bad guys could be there. So beneficial uses for fermenters. I mean, you guys know this. You don't need me to tell you this. You guys know you like to eat cheese. I know you do. You like to eat bread. Maybe you drink some beer or some wine, or maybe you eat kimchi. I'm not a fan. Or you drink kombucha. I'm not a fan. Or you eat pickles or like sauerkraut. I like both of those things. So, or yogurt. All of these things involve bacteria that would be fermenting carbohydrates, sugars in something and turning it into via fermentation, because they aren't going through aerobic respiration. They don't have oxygen available. So they just go through glycolysis. And when they get to pyruvic acid, they just turn that pyruvic acid into alcohol, ethanol, like we drink, or acid, like organic acids like lactic acid. And then often carbon dioxide can be a product of that as well. Not always, but it can be. So a lot of organisms can do that in different ways, but that's how they deal with just only doing glycolysis and not moving on to the other steps involved in aerobic respiration. That obviously we use the alcohol made from that, and we can use the acids made from that. And we don't even just use it for foods, we can also use it for industry as well, like producing organic acids for use in industry, for example. So freezing microbes will not kill them. Freezing microbes will not kill them. In general, just assume no microbes can be killed by freezing. There are some that can, but do not rely on that for food safety, please. It will preserve your food, stop most microbes from growing, but it's not going to kill most of them. All right. So when you get it back out of the freezer and you thaw it out, don't consider it sterile or something. It's not true. Okay, next slide. Chapter 10, not particularly difficult. Restriction endonucleases. These are enzymes that are from bacteria that in nature, they use them as defensive against foreign DNA, foreign genetic material that they don't want there. So that's from bacteria. And how do they work? They recognize palindromic sequences that are 4 to 10 nucleotide bases long. And so they read the same forward as they do back on the anti-parallel strand, complementary to them. And then usually each half also sort of matches up, in case you haven't noticed that. But so it's even like almost double palindrome. But whatever, they recognize palindromic sequences, they cut at those spots, and they leave sticky overhanging ends that allow us to cut our own target DNA of whatever, cut it with the same enzyme, to create the same ends. And we can take our DNA and it'll stick in with that bacterial plasmid or whatever. And then we can seal it up with DNA ligase, and then that will allow us to create, genetically engineered organism that can produce whatever protein we want it to, essentially, when we do that. So cool stuff. So exactly, when you stick 2 pieces of DNA together, you have to seal it. The thing we use is DNA ligase. Like I just said, the bonds that are being made are called phosphodiester bonds. That actually spelled it correctly in that. Okay. Sorry, it's just fascinating to me. I've never used it before. Gel electrophoresis. That's just going to be using an agarose gel. So I did not say agar, I said agarose. So it's a different thing, okay? It is based on a sugar that may have been derived from agar, but it is not agar. It is agarose. Anyways, what we do is we make these little gels and basically put DNA at one end, our sample at one end, in a solution, a buffer. You guys know what buffers are. They maintain pH and electronegativity of the environment and all of that stuff. So we pop our gel into the buffer, put our sample into the wells, turn on the electricity, and because of the charge of DNA being negative charge, it's going to move down the gel, through the gel. And the smaller pieces are going to move faster because the big pieces get caught up within the gel because they're bigger. So it's like trying to muscle your way through a crowd. The littler guys are able to sneak through maybe a little bit quicker. So that is what's going on with gel electrophoresis, and we can separate out DNA fragments based on their size using that. Nucleic acid hybridization is taking a little piece of maybe a gene that you're trying to look for in the bacteria that you just created or some other sample that you have, but you take a little piece of that, single-stranded, and you take your, I don't know, bacterial plate, and you put a little membrane on it to pick up all the cells from your colonies, and then treat it to cause the DNA to separate apart into single strands. Put your little probe on there, your single-stranded probe for the gene of interest, and then you can use certain markers or radioactivity, whatever you're using, to determine which colonies on there contained the gene of interest. So that would allow you to pick out of a population which group might have the gene that you're interested in. Maybe it was something that was, you're trying to put a new gene into bacteria so that it'll make a new protein or something. And if you're having a hard time with doing that, you might use nucleic acid hybridization to be sure that the bacteria that you're moving ahead with actually has the gene that you're interested in. What are the major components and steps in PCR? PCR has 3 steps, okay? It has denaturation, which means splitting it apart. We use heat to do that. So split the 2 DNA strands. By the way, this is for DNA, in case you guys didn't know that. Polymerase chain reaction, PCR, amplifies DNA. It makes copies of DNA very quickly. You can go from like 1 copy to billions in a very short amount of time. Are we barking now? Is that what's happening? Are we barking? Anyways. Right, so, right, denaturing the DNA strands, the double-stranded helix, and heating it up to cause them to separate. That separates the actual hydrogen bonds that are holding those together. The A that's bound to the T, or the G that's bound to the C, those guys get pulled, those are held together typically by hydrogen bonds. So the heat is going to separate that, actually literally separating the DNA strand. All right, so then we have the next step, which is anneal. Anneal, sorry, that translation. Anneal, A-N-N-E-A-L. Annealing is where we're going to be adding our primers, and they're going to be designed for whatever portion of the DNA that you're more interested in, because you don't want to wait for the entire genome to get copied. So you're going to copy the area that represents that there's E. coli here, because this gene is found in E. coli, right? So you add your little primers that are indicative of whatever you're looking for, and cool it down a little bit. The primers will sit onto their little matching points on those, and then we will have elongation, which is the 3rd step, where the polymerase, which in this case we have a heat stable polymerase, and it will create elongation on each of the strands that we've separated apart. So now we've gone from 1 double strand to 2 double strands, and then we'll keep separating and keep copying, keep separating and keep copying, and so on and so forth, till we have billions of copies. And that is how PCR works. The major components, well, obviously you need your template. So that's going to be whatever your sample is. You need your primers. You need a buffer, which is going to provide like things like magnesium and things for the polymerase. There it is, the polymerase to be able to function. And it includes the nucleotides, the free nucleotides that we're going to be using up during the process to build new strands. And then we also have to have the polymerase, which will be heat stable from bacteria that are typically living in deep vents or something like that. Righty. Cloning is essentially going to be taking genes and manipulating them through things like PCR and stuff like that, making changes in them and potentially even putting them into new organisms or back into the original organism, depending on what you're doing. But either way, if it's been manipulated with this genetic engineering stuff, then we will consider that to be cloning. So GMOs are genetically modified organisms. So this is anytime you have any organism that you've put a gene from another organism into it. So that is also considered transgenic. And when we have that, that'd be like E. coli, that we put a human gene into it that produced insulin, which is a real thing. That's how we get insulin now. So that is a GMO. That is a transgenic organism. How are plasmids useful in genetic engineering? I sort of explained that kind of throughout all of this, I feel like. But remember, plasmids are small circular bits of extra chromosomal DNA that eukaryotes can have them, but we're primarily interested in the bacteria. So they're extra and they're not required for survival. So what we can do is we can put genes of interest, and using our restriction endonucleases, make cuts and insert genes that we're interested in and splice them in to the plasmid, and then put that plasmid back into the bacteria, and it will start producing the gene that we want it to. Gotta remember though, that when we do this with bacteria, that if you're putting a human gene in there, that you have to have already removed the introns. If you don't know what I'm talking about, then maybe go review your genetics chapter. Right, so I mean, I feel like that explains why they'd be important for genetic engineering. We can put pretty much whatever gene we want into these organisms, and then, you know, reproduce whatever the product is via that mechanism. And bacteria grow a lot faster than eukaryotic cells do, so that's a pretty useful tool. Gene therapy and the challenges. Gene therapy refers to essentially putting a gene that works properly from a healthy individual into an individual who has defects in that gene, and permanently. So we can actually change the cells of their body this way. Now, how do we manage that? One of the most popular ways is to utilize a virus that would be like HIV, that is able to incorporate its genome into the host genome. Come here. I know, you're small. You're very cute. I know. Incorporating it into the host genome, and then it would become permanent if we could, you know, use that. Well, we take out the genes for HIV and replace them with whatever gene that person is having a problem with, and then mix it in maybe with their bone marrow, for example, and then put their bone marrow back into them, and then their bone marrow becomes, you know, infused in their body and incorporated back in their body, and it grows and it spreads and whatever, and supposedly will spread this throughout the entire body. It is being used. There are, I think, 13 diseases that we treat right now with gene therapy that this is working for. You would hope it would be more though, right? So what are the challenges here? The problem is the body sees those viruses as being foreign and wants to attack it. Really small. And then, you know, we'll attack that, or maybe it just doesn't get incorporated into enough of the cells, or enough of the cells survive in order to pass it on to other tissues to be maintained within the actual body permanently. Or there might be negative side effects from things that you didn't anticipate, because you can't anticipate everything. All that stuff can cause problems. So that's the issue with gene therapy. All right, microbial control. This part is just not particularly exciting to me, but we have definitely got to talk about it. It is important as far as microbiology is concerned. So yeah, we will go ahead and actually do that like this. Better. All right, compare and contrast sterilization, disinfection, antisepsis, and decontamination. Sterilization, removing all microbial life. Disinfection, removing most of the microbial life, but not all of it. So often endospores are left behind. Antisepsis is, so disinfection, that's going to be inanimate objects. If you're doing this on animate objects like skin, that's antisepsis. So disinfection and antisepsis, same thing, except disinfection is on inanimate objects. Antisepsis is on animate surfaces. Decontamination, this can be removal of microbes from things mechanically. So it could be, doesn't matter if it's skin or if it is inanimate. It could be either one, but if you're using mechanical means to remove it, that would be considered decontamination. The difference between microbicidal and microbistatic, you guys probably already know this, right? Kirby Bauer, you guys learned this. So microbicidal, we have actual killing of the microbes, and microbistatic, we have inhibition of the growth, but not necessarily killing, right? So if we remove it, they could start growing again potentially. So typical targets for antimicrobial agents, we have the cell wall, the cell membrane, proteins, nucleic acids. So it's going to be the same for whenever we actually get into the drugs, but we're going to add one more, which will be folic acid. So it'll be 5 for those, but for this, it's just 4. What effects do detergents and alcohols have? They often can disrupt the membranes and the cell walls of these organisms to make them weaker, and therefore helping other agents typically do a better job of killing cells. Now, alcohol is a little bit different because it not only disrupts the membranes, but will also be involved in the cell walls, will also be involved in denaturation of the proteins. So we need a little bit of water in order for that to happen. We need some water in addition to the alcohol to cause protein coagulation and therefore inability of the proteins in that cell to function any longer. So that leads to denaturation of those proteins. Yeah, alcohols will do that. So they're pretty useful in that regard, but you do need to have water. You can't have 100% ethanol or 100% isopropyl alcohol. It's not useful. You need water there to help with that whole effect. So what property of surfactants help them function? Surfactants are, you can think of soaps, okay? Think of sudsing and stuff like that. One of the surfactants are the quats, which is question number 16, by the way. Quats are quaternary ammonia compounds, and they are surfactants. They're chemical surfactants, and they control microbes this way. So I'm not going to re-answer this question when I get to the bottom. They have a charged head and a long hydrophobic carbon-hydrogen chain, and look a lot like maybe a phospholipid would look with that charged head and the long chain. So they can sort of what we call intercalate or squeeze between the phospholipids and the actual membranes. And then that destabilizes the membranes and allows for them to open up. And that's more useful when we're trying to have other compounds and chemicals get into the cell to kill it proper. So it makes other things more effective. We often combine this with chlorhexidine in surgical scrubs. So we'll have a surfactant, it's often a quaternary ammonia compound that will combine with the chlorhexidine, and then you scrub and we have mechanical removal of organisms when we're scrubbing. We have the surfactants to help loosen up the actual cell membranes to allow the chlorhexidines to get in and do their job. Chlorhexidines are phenolics, so they're based off of phenol. That's how they function. So chlorhexidine is pretty useful in hospital care settings. All right, next thing, the most common methods for microbial control what is one of the most common methods for microbial control that targets by denaturation, and what 2 forms is it used in, and which one is more effective? We're talking about heat here. So we have either moist heat or dry heat. Moist heat is more effective. It's sort of like what we were just talking about with the alcohol. The moisture helps with denaturing the proteins and being more effective as a sterilizing agent. So, so that's moist heat or dry heat, and the moist is more effective. Yes. Thermal death versus thermal death point. Thermal death time versus thermal death point. Thermal death time is the amount of time that it would take to kill all of the microbes in a sample using your chemical of interest. Whereas thermal death point will be, and the first one at a certain temperature, right? Is using your chemical of interest or means of killing microbes, whatever you're looking at. Could be autoclave, it could be, some sort of cleaner that you're looking at, whatever it is. The thermal death point is the temperature, the minimum temperature that you need to reach with that autoclave or whatever it is that you're testing, the minimum temperature that you have to be doing that at in order to kill all of the microbes in 10 minutes. So that is the thermal death point. So what is an autoclave and why is it so effective? Please tell me that by now you know what an autoclave is. An autoclave uses pressure, steam, and heat in order to sterilize things. So that's what it does. It pressurizes a chamber, helps it reach the temperature that you're looking for, usually 121 degrees Celsius faster. And then we'll maintain that and add in steam in order to effectively sterilize things. So because it uses all of those things all together, especially with that moist heat, right? That's what makes it so effective. What benefit does pasteurization have? It allows us to maintain the flavor and appearance of a liquid without overheating and denaturing things and causing flavor changes and all of this stuff. We can still remove the pathogens from there. However, it won't remove all of the microbes, so it will not be sterile. And this is just cycling between hot and cold and repeatedly over and over again until you've destroyed whatever pathogens you're trying to destroy. Boiling, why don't we use it to sterilize? We don't use boiling to sterilize because it doesn't sterilize. Endospores are more than happy to survive boiling. So you can't sterilize anything with it. You can disinfect things with it, but the problem is as soon as you take it out of boiling water and put it back into the environment and it's cooled down, then it's pretty easy for things to get, recontaminated. So it's not super useful unless it is like, you're trying to maintain cleanliness in a home while somebody is ill or something like that. It can help with that. Better than nothing, right? Explain the difference between ionizing and non-ionizing radiation and which is more effective. Ionizing radiation, we expose something to ionizing radiation and it causes the electrons, the orbital electrons on the outside of that to be ejected, essentially. And this can cause massive damage to the molecules in whatever substance you're treating. So even including bacteria that might be on the surface. So this can cause massive damage in a lot of the molecules of the cell, but particularly almost irreparable damage to DNA and things like that. Non-ionizing radiation. So when we are ejecting the electrons from orbitals, of course, we're creating ions. I hope you guys remember that from chemistry. So that's what's going on there. Anyways, let me check and see if people are in here. I don't think they are. Nope, I'm still by myself. Okay. Non-ionizing radiation is like UV radiation, ultraviolet radiation. It doesn't penetrate barriers. Barriers are pretty protective as far as keeping it from going to areas that it shouldn't be. It causes in DNA, it causes pyrimidine molecules to create bonds to one another when they're next to each other. So thymine dimers is the typical example that we give. Two T's are next to each other on the same strand of DNA. And they will no longer bind across to the A's that they would be associated with in the complementary strand. So they're bound to each other now. So whenever we go to replicate that cell, there would be a problem being able to replicate it due to that defect of the T bound to the T. So we have to have a means of correcting that if we're going to be exposed to ultraviolet radiation. This does not sterilize. So this is not typically used for sterilization, but can be used in combination with other things to reach for sterilization. So that's non-ionizing. Filtration, you can literally have a small enough pore in your filter, whatever paper you're filtering through, whatever it is, you can have a small enough pore to remove all known microbes, even viruses. You can filter them out. It's unlikely that most of the stuff that you're using at typical home setting would remove every single microbe ever and like completely sterilize the air, for example, because you can use filtration on the air in addition to fluids. It doesn't just have to be fluids, right? So if you have a HEPA filter on like your vacuum cleaner, that's filtering the air. It's probably not sterile. You can achieve sterilization with filtration, though it's just going to take small enough filter holes to trap everything. Osmotic pressure, that is referring to like packing something with salt and having that super hypertonic environment outside of the cell that is going to draw moisture out of the cell. That whole pressure to pull out the water from the cell, that's called osmotic pressure. So packing in salt or packing in sugar, you can also do it in sugar. It has the same effect. Still is a solute on the outside of the cell that you can have a high concentration of. So we do pack things in salt or in sugar, especially like if you're creating preserves or something like that. We can use high amounts of sugar in order to preserve like your fruits. So these things can help with food preservation that way, creating that osmotic pressure, that hypertonic environment. What are some examples of halogen-based agents? I mean, I feel like iodine. So if you've ever heard of betadine, that's got iodine in it. That's a halogen, iodine is. We don't use it as much anymore, but you've probably seen it. That sort of orangey, brownish, yellowish, all at the same time, but it stains pretty badly. And it's not always used properly in healthcare settings. So there's a problem there. And some people are allergic to it. So they prefer, in general, chlorhexidine nowadays. But you can be allergic to that too. Then we have fluoride that's in your toothpaste or in your water. And then we have chlorine, chlorine-based compounds like bleach. So bleach is sodium hypochlorite, and it is a pretty effective halogen-based cleaning product. And those chlorine-based compounds are, especially the hypochlorite type compounds, when used correctly, are actually quite effective at sterilization. So we can actually remove all microbes. What principle does using hydrogen peroxide as a microbial control agent work off of? Hopefully you have been paying attention in the past few slides. So brought it up like 4 times, but oxygen, toxic oxygen molecules. So hydrogen peroxide is one of those molecules that can cause damage to cells. And so those cells that would be maybe not able to deal with hydrogen peroxide, which the enzyme that does that is called catalase. If they don't possess catalase and we expose them to hydrogen peroxide, then that is going to, kill them. So that's what we use that for. And you can actually sterilize things with that as well if you're using a high enough concentration of hydrogen peroxide. Hydrogen peroxide, even at the highest concentrations, will kill just about anything, no matter how much catalase you're producing. If you've ever put hydrogen peroxide on a cut and seen it foam up, bubbles, 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 that is catalase doing its job, converting that hydrogen peroxide just into water and oxygen. And the oxygen is what causes the bubbles. So we will see that in our bacteria when we do that test on our unknowns later in the semester. All right, quats. Oh yeah, we already talked about this. Okay, good. I already talked about how quats or quaternary ammonia compounds are surfactants. Cool. So that is that for this week. And if you guys have any questions, of course, contact me, let me know. And I know the exam is coming up, so please, as you're studying, if you need help with anything, don't hesitate to contact me.